Why is EEG data interpretation complex? Imagine trying to listen to a single conversation in a crowded room filled with voices overlapping. That's similar to what interpreting electroencephalography data feels like. EEG records electrical signals from the brain, but these signals are highly dynamic and multidimensional. They come from many different neurons firing at once, and their signals blend together, making it tough to tell which activity belongs to which part of the brain. One big challenge is signal quality. The data often gets mixed with noise from muscle movements, eye blinks, or even electrical interference from the environment. These unwanted signals can make the data hard to read. To fix this, specialists use advanced filtering and artifact removal techniques to clean up the signals before analysis. Another difficulty comes from the way EEG signals are collected. Electrodes placed on the scalp pick up electrical activity that has traveled through layers of tissue, skull, and skin. This process distorts and weakens the signals, so what's recorded isn't a direct map of brain activity. This problem is called the inverse problem, and solving it requires complex mathematical models and source localization methods to estimate where in the brain the signals originate. EEG patterns also vary a lot between different people and even within the same person over time. Factors like age, mental state, health, and brain structure influence these patterns. Because of this, creating universal standards for interpretation is difficult. Instead, personalized approaches are often needed to get accurate results. The data collected from EEG involves many channels, sometimes over 100, each recording at high speeds. This creates massive datasets that are difficult to analyze manually. To handle this, researchers use computational tools and machine learning algorithms to find meaningful patterns and reduce the data's complexity. Standardization is another hurdle. Different labs and clinics might use different electrode placements, equipment, or settings, which can lead to inconsistent results. This inconsistency makes it harder to compare data across studies or patients. Understanding how different brain regions connect and communicate adds another layer of complexity. Analyzing these connections from scalp recordings is tricky because signals can appear connected when they are not, leading to spurious correlations. In neurotechnology focused on brain mapping and diagnostics, these challenges are tackled with advanced signal processing, machine learning, and visualization tools. For example, transforming raw EEG signals into images or time-frequency maps can reveal hidden patterns. Machine learning models help classify brain states or detect disorders, but they need to be carefully designed to manage variability and limited data. Practically, interpreting EEG data helps diagnose conditions like epilepsy, sleep disorders, and Parkinson's disease. It also plays a role in brain-computer interfaces that allow direct communication between the brain and external devices. Improving how we interpret EEG signals makes these applications more accurate and reliable, leading to better health outcomes and innovations in neurotechnology. All these factors, noise, distortion, variability, and data complexity, make EEG data interpretation a challenging puzzle. But with the right tools and techniques, scientists and clinicians can piece together a clearer picture of brain activity and its many mysteries.